I am Erdoğan Eşref Hakki from Selçuk University in Central Anatolia, Turkey. Before starting on my talk on the potential of extremophiles for agriculture under adverse conditions, I would like to thank to the organizers for inviting me as a speaker for the first International Soil Science Conference. I am attending from Konya, the central part of Turkey, the largest province and the agricultural province of Turkey. Actually, Konya is a very old establishment. Its history goes back to 10,000 years. The first city settlement on Earth is in Konya, namely Çatalhöyük. And there are some primers in terms of agricultural practices also. Most important for us, we had the chance to analyze ancient DNA from excavations of Çatalhöyük going back 8,400 years, and some implication for the Neolithic origin of agriculture at this region. But the soil in the region is tired. We suffer some sort of wind erosion. The land is dry, about 300 millimeter precipitation annually, and desertification is a threat that hit actually the region during 60s, and there were some good example of fighting with this issue during that time. Our research is mainly related with salinity. The salt lake seen with the arrow is the largest in Turkey, and around that region, not only saline soil, but at the same time highly boron toxicity issue is prevalent, and about 23% of all Turkey is actually the land is suffering from boron toxicity. The orange and red colors indicate this. In Turkey, we call the October Ekim, which means in our language sowing, because we were sowing wheat and cereals during October, but not anymore. Last year we were sowing during December, and this period extends. There is a shift of the season, and even during January the weather was quite shiny, quite good for sowing. So we have new problems, and climate change an issue which we were skeptic about this during early stages of our professional life, but recently we realized that humanity is approaching an unprecedented catastrophe. During the last decade, different regions of the planet have been severely impacted by exceptional weather and climate extremes, including heat waves, hurricanes, drought, and heavy precipitation. So this is frequent extreme events contribute to complex abiotic stress conditions that endanger crop yields and food security. And this is not coming as a single stress, but multiple stresses in combination, also an important issue to be considered today, like heat and drought, salinity and drought, water lodging and heat, and looking for the projected impact, combined stresses could reduce, for instance, major crop yields by up to 7% per 1 degrees of global warming. When we consider this issue, usually the first thing we think is the use of biodiversity for alleviating these effects. And two foundational strategies are under consideration: harnessing intraspecific variation through advanced phenotyping and genomic assisted breeding. And the second one is exploiting wide relative germplasm via interspecific hybridization and trait integration. And now we consider boron toxicity, salinity, and drought, and many different disciplines should work together in coordination so that we have some solution for this important issue. And I will talk our approach on this in shortly. So while gene discovery is important, precision breeding tools like adapting new technologies like CRISPR-Cas9 is important, especially for the future applications. And advanced screening methods may be very helpful, high throughput phenotyping systems to be adapted may be important to solve this problem. And considering precision breeding, wild gene mining example may come from different crop species like tomato, rice, wheat. Our experience is mostly on wheat, and we usually apply marker-assisted breeding, and usually we deal with abiotic stresses. But there are other species, also successful applications like bean traits that improved common bean drought tolerance up to 40%. And there are also some barriers on new technologies like CRISPR, and also linkage drug, genetic complexity, important issues to consider. And these are challenges, and the enabling technologies yet suggests some important improvement, and research notes that high throughput phenotyping is one of the important topics to be dealt with. I'm going to summarize our own experiences, which started during 1990s by analyzing our soil, our plants, our water, and we determined the toxicity and deficiency rates in our regions. That was also followed by a national project, and we now have a map of boron in Anatolia, and this provides a good map for our future work. We started with trials in different regions using different crops species. Many researchers in our institute contribute to this research, and shortly we started shifting to the controlled environments in greenhouse experiments using again many different crop species and determine the situation when uh, especially boron is considered. So we also applied different elements in our research and like salinity and boron toxicity or boron iron interactions among other studies we worked during this period. Interestingly, some applications like certain amount of boron application alleviates the salinity stress response, and this is also followed by other research within country or abroad. And we started thinking about more extreme conditions and mining areas. We search all around South Central Anatolia, and we determine different plant species. Among them, one grass, the Puccinella, is here. That is very important, but there are a few more species. I'm not gonna talk. We started with some physiological biochemical analysis using this species that is highly tolerant to boron toxicity. So we shifted to more closely related to wheat species also like Brachypodium or even some crops like barley. We tested different plant species with much reduced amount of boron. Then we started thinking about during 2010 physiological, anatomical, and ionomic or some gene expression studies during that period. Many projects and many theses we completed during this time, and we focused on some, for instance, novel boron efflux transporters coming from these plants grown in mining areas. And this RNA-seq data provides many such new genes that are to be discussed. Now, as you may understand, we are focusing on gene pools, first national, international varieties or landraces or close relatives. We don't have the chance to deal with transgenic at least during that time in Turkey. So our gene pool is basically supported by landraces or relatives, and we started transfer of NAX genes and boron toxicity tolerance genes. These are coming from CSIRO in Australia. We collaborate with the Moons group, and this was the first generation. We included also boron toxicity tolerance genes coming from a tolerant Turkish old variety. But when we progressed, we started thinking second generation applications also. We started thinking to include some grain protein or iron zinc concentration improving genes like GPC B1. Now this work is in progress. Key research gaps include epistatic interaction between introgressed wide alleles and elite backgrounds, tissue-specific expression of wild stress response genes, and trade-off between resilience and agronomic performance. Soil salinization affects quite a great amount of irrigated wheatland, so quite a high amount of losses, but at least we have some tools to deal with, like wild genetic resources from, for instance, NAX2 locus from Aegilops tauschii genome progenitor, or some others also genes may be utilized coming from different sources. As I try to explain, new techniques are promising, but marker-assisted selection is also a good tool to consider this kind of research because of the regulation issues to be dealt still while using CRISPR technology. So phenotyping innovations and high throughput platforms provide important information, but technical challenges like NAX linkage with reduced grain size up to 7% when pleiotrophy is considered, and regulatory barriers are important issues, as I said. We use 16 combinations. 10 of them were bred wheat, the remaining durum wheat, and now they are enhanced lines, backcross self, and we collect some physiological data. Different students from our groups working on this and collecting data nowadays. So second generation applications using also quality trait, as I mentioned, coming from some of the PhD students' studies that are in progress right now. And next, we started focusing on the rhizosphere of some especially important tolerant species. The best one is the Buccinella distant, as I mentioned. It is highly tolerant to not only boron toxicity, but also salinity and drought. It tolerates quite important plant species. Actually, when microbiome is considered, we have quite good knowledge nowadays coming especially from human health, but linkages establishing between plants microbe and human health are becoming more and more important. And we in our research focus on soil microbiome, more specifically rhizosphere microbiome. And when we consider sustainability, we believe this is an important issue. Actually, at the beginning of our career, we were thinking that the travel to Mars was only a topic for few researchers from some developed countries, but nowadays we are in different stage. We are thinking permanent colonies there and exciting topics of biology. Once we were having biology education, capable of living at highly extreme conditions nowadays started to be um, utilized from industry or laboratories, or we have possibly the chance to get more benefit in agricultural systems as well. For instance, in laboratories, we are using enzymes from archaebacteria, but fungi also, and some few more eukaryotes as well have some promises. But also some very familiar microorganisms like Bacillus subtilis that provide important tolerance or against different extreme conditions, including high pressure, UV radiation, salinity, or temperature, heavy metals. These are important. There are some exceptional organisms also, like the tardigrade, which is a polyextremophilic organism that can tolerate many different stress conditions. Research nowadays focus on deciphering its system, how the DNA damage is repaired successfully, and how this organism is protected, and how we can use these genes to make more tolerant and more adapted to different stress conditions or multiple stress conditions places. So we can think about these engineered microorganisms nowadays. Now, basically, we deal with prokaryotes actually, and we use different extreme conditions, and many disciplines are needed to work in coordination, starting by ecology, diversity, taxonomy, evolution, astrobiology all together. And luckily, we have right now high throughput isolation and characterization systems, so we can use this information for the outcomes like novel species strains, efficient novel enzymes, metabolism for toxic compound degradation, new types of important drugs, novel astrobiological model using this system, novel compounds to improve some industrial processes, as well as the improvement of agriculture, which is the main interest of our talk. In our own research, we focused on the rhizosphere of Puccinella distance, and we isolated and characterized microorganisms that are dominating the rhizosphere under these highly toxic conditions, and we determined actually three species. One is the Pseudomonas extremorientalis, the other one is Olibacter soli, and the third one is Pseudomonas poia. We focus especially these organisms in our research, and we started using this organism testing different plant species, different stress conditions, including drought, including boron toxicity, including salinity, and some promising results are started to come from these studies using different crops from onion to strawberries, from lettuce to wheat, and at the same time we started testing our organism in the presence of some commercial also uh, formulations recently, and some good promising results. Here are some more example of our trials, some ongoing studies like the nanoplastic application in the presence of drought conditions, and application of specific microorganisms to alleviate the effect, or we don't know yet what we will get as output from this research. And we also focus on some industrial crops. Recently, cannabis is one of them, and we started some basic studies by the application of microorganisms, some secondary metabolite, cannabinoid profile, or some different metabolize profiles were drastically changed, and these preliminary studies are promising, and we will soon thinking to apply in our region. This crop is also started to be cultivated for different purposes. So combined boron, salinity, drought, important topics nowadays in our research group. We get benefit from biodiversity using wild species, or genes coming from relatives, like NAX genes coming from Triticum monococcum, in collaboration with Australia's Cyro Center, and some Landers's local having this boron toxicity tolerance gene. And we focused recently on microbiome research in our group, and at the same time try to understand the mechanism of these tolerances. And thank you for your attention. Thank you.